I'll go ahead and warn you, this is going to be the longest video of this chapter. But here we're looking at all the catabolic pathways. We have aerobic respiration here with oxygen as the final acceptor, and we can theoretically make up to 38 ATPs. And you see glycolysis, the Krebs cycle, and the electron transport chain are all part of that. Anaerobic respiration is the one in the middle, where we have glycolysis and the Krebs cycle and the electron transport chain. But the electron acceptors are non-oxygen, things like sulfates and nitrates and carbonates. So how many ATPs are made is going to vary with the microbe. The last process, fermentation, is only glycolysis. And it's an organic molecule as the final electron acceptor, like pyruvate or a type of alcohol or something like that. So it have a maximum production of 2 ATPs there. Now, aerobic respiration, we've already said, is the most productive one. In 103, we refer to it as cellular respiration. It's the same thing, okay? This is a series of enzyme-catalyzed reactions where the electrons are transferred from the fuel molecules, the glucose, to the oxygen as the final acceptor. And it involves, as we've said previously, glycolysis, the Krebs cycle, and the electron transport chain. With glycolysis, we take a 6-carbon glucose, and it's oxidized. And we split it into 2 molecules of pyruvic acid, and they each have 3 carbons. So we've taken 6 carbon and split it, and we have 2 3-carbon pyruvic acid. And we make NADH, carrying those electrons. Okay? In the Krebs cycle, we take the pyruvic acid, and we make 3 carbon dioxide molecules. And NADH, excuse me, and FADH2 are made. And these are also carrying electrons. And remember, electrons have energy in them. Okay? The electron transport chain is going to accept those electrons from the NADH and the FADH2. And then it's going to make energy by a series of redox reactions called oxidative phosphorylation. So in looking at aerobic respiration, starts with glycolysis. We take glucose and we split it into 2 pyruvates. And you see we've made a little bit of ATP, and we put the electrons in the carriers, the NADH. Alright, then the 2 pyruvates are going to go into the Krebs cycle. We're going to give off some carbon dioxide, and some NADH and some FADH are going to load up with electrons as well. And then those carriers, the FADH2 and the NADH, from these other 2 reactions are going to go into the electron transport chain, and here oxygen is going to accept those. So we can make up to 38 ATPs here. Now, let me go back and show you that again. The 2 pyruvates is what links it to the Krebs cycle, okay? That are made when glycolysis splits glucose into 2 pyruvates. So that's the link. And when it's split, it converts it to acetyl coenzyme A. That means that we're going to reduce the NADH and or NAD and make the NADH, and remove the carbon dioxide molecule with it, which is decarboxylation. Okay? So we have removed the carbon dioxide, and here's the NADH. Okay? So we reduced the NAD to make the NADH, and we removed the carbon dioxide. Decarboxylation. it goes around twice. One, because you've got two pyruvates, it's got to go twice, one for each pyruvate. And every time it turns, it makes one ATP. So it turns twice, so it makes two ATPs. Go back and show it to you again. Okay, the two pyruvates give off the carbon dioxide, and then we have the NADH. It's converted into acetyl coenzyme A. We're going to turn in this Krebs cycle, and it's going to turn twice because we have two pyruvates, so it's got to go around once for each time. We're going to produce carbon dioxide, two ATPs, and then we're going to harvest the NADH and the FADH2 with the electrons that are given off. Went the wrong way, sorry. The final processing of electrons and hydrogen is the main thing that generates ATP, up to 34 ATP molecules in this electron transport chain. We've got these redox carriers that receive all those electrons that have been brought to it from the NADH and the FADH2. And they go through the electron transport chain, and the energy is released and captured and then used by ATP synthase, which is an enzyme, to make ATP. We look at the electron transport and chemiosmosis. That carrier, that NADH, is going to transfer the electrons to the carriers. And the carriers in the electron transport are called cytochrome molecules. And here you have two electrons are going to combine with two hydrogen ions, which are just protons, and an oxygen atom, because remember oxygen is the final acceptor, and they're going to form water. So this electron transport also involves pumping the hydrogen ions or the protons across the membrane into the intermembrane space by three carrier complexes. When we talk about chemiosmosis, these are the electron transport carriers that shuttle the electrons. They actively pump those hydrogen ions, those protons, across the membrane, and that causes a gradient of hydrogen ions. And they actually pump it into the intermembrane space of prokaryotes, and that causes that gradient between the intermembrane space and the matrix. And that creates an electrical potential. So that drives the movement of the electrons or the protons through from that intermembrane space through the ATP synthase that's in the membrane, and that causes the production of ATP from ADP and inorganic phosphate. So this is what it looks like in the prokaryotes, all right? You remember we have this periplasmic space in here. Here's our electrons in our membrane, and you see we've got all these hydrogen ions in here, and the electrons are going to move through this transport chain here, but the hydrogen ions, the protons, get pumped through into this periplasmic space, this intermembrane space, and we end up with a gradient. We have more of them up here than we do down here, and so they will move through this ATP synthase, this enzyme complex, and when they do that, we take ADP and add a phosphate to it to make ATP. Now, we've said oxygen is the final acceptor. It accepts two electrons from the electron transport and picks up two hydrogen ions from the solution and makes a molecule of water. So with aerobic metabolism, we yield up to 38 ATP molecules. So if we look up here in the right column, the net output summary, you can see in glycolysis here in the yellow, we make 2 ATPs. 2 NADHs pick up the electrons, and 2 pyruvic acids are made from the glucose. But then we have this little transitional period where the pyruvic acid is converted into the acetyl coenzyme A, and then it goes into the Krebs cycle. And in the Krebs cycle, in the green, we're going to make it goes around twice. We're going to make 4 carbon dioxides, 6 NADHs pick up electrons. We're going to make 2 ATPs because it goes around twice. And we've got 2 FADH2s picking up electrons. Then it goes into the electron transport, and oxidative phosphorylation happens, and we make 34 ATPs here and 6 waters. So 34 plus 2 plus 2 equals 38. That's the possible yield. What part of aerobic respiration releases carbon dioxide? I only mentioned it during one part.